All right, thank you, Tommy. So I understand many of the folks in this room have a pretty good background in things like machine learning and computation more broadly, but maybe less of a background in neuroscience. I think my job was to try to give you guys some feel for what goes on in some aspects of computational vision. The talk I put together is, first half is going to talk about some review of the visual system, and then the second half will get more into some specifics of how at least my lab is applying some of the things that I think you guys are learning about to the problem of visual object recognition. But more broadly, you can think of what we're after. When I say we, I mean all of us, I hope, in this room. Many of us at least interested in vision is computational algorithms that can explain challenging visual tasks, at least how the brain executes those tasks. And I want to sort of start by, my slides will advance, they're not doing that. Sorry. There we go. So this is a slide actually that I stole from Tommy at one point, but it's just to illustrate generally the problem of vision. So you all look at this and you quickly do things like identify that these are cars, these are people, that the signs are buildings. You can maybe estimate that this is a walkable surface. You probably even start doing this predicting what these people are going to do next, whether it's safe to cross the street. You do a lot of that very, very quickly with this whole scene here. So there's a lot of visual tasks there. Now, I want to sort of say that my lab actually works on, collaborates with Defense Department on many things, and there's something I thought it would be fun to share with this group that is still somewhat classified, if you will. There's not fully classified, but some of it is not, I can't tell you the details of yet. But there is this sort of computational system that has basically 100 billion computing elements, solves problems not solved by any previous machine. Actually, this doesn't look like it, but it only requires about 20 watts of power. And again, I said it's still classified. And so you probably know what I'm doing here. I'm not talking about this thing. I'm talking about that thing. Okay, so there's this amazing set of algorithms in here. That's how we think about it, that we'd like to reverse engineer or extract from the brain. And I know many of you are probably interested in those things as well. And so let me tell you that, again, what I'm going to talk about today is the problem of visual object recognition. And this is how it's operationalized in computer vision, is to be able to at least put bounding boxes around this and be able to identify things. Now, this is maybe not exactly how our visual system does it, but at least it's an operational definition of the problem. And I'll be using a definition that's very close to that. So first I want to back up and talk about why study object recognition in the brain more broadly. And it's because these representations that our brain is computing from the visual world are really the substrate of higher level cognition. I mentioned things in the last slide, like being able to predict where a person's going to go next, whether that car is going to hit you. You can't do that unless you first estimate those items in the environment and their states and perhaps their future states. So they underlie, again, things like your memory, what things you're going to lay down in memory, your value judgments, again, decision and actions. And if you're building robots and you need these kind of, what you will, front-end systems to do things like obstacle avoidance, navigation, danger avoidance, etc. I won't read all those. Okay. So again, why study this? Well, the other reason that I want to point out is that, as I mentioned, it's already a challenging computational problem. So the goal, I want to point out this slide from, this is from 1966 from MIT. There's a summer vision project here. I used to think this was an apocryphal story, but Mike Tarr actually then gave me this document that there was a goal in the summer project to have some undergraduates put together a system. This was in the sort of heyday of early AI, sort of first round of AI, that we would solve object identification and would name objects by matching the vocabulary of known objects. There were some goals for July and August, but the goal is to essentially solve this problem by the end of the summer. That was 50 years ago. I would say now we're almost 60 years ago. I think we're close to starting to solve this problem, but I want to point out that we had an intuition that some of these things were easy because our own brains do them quite well, yet it turned out, as many of you already know, that this is a very, very hard problem. If you're ever interested in this document, it's really a fascinating sort of Mad Men, like the objects they pick are like cigarette containers and things that we would not look around our offices and see, but that's what they describe. Okay, let me sort of then put that in context. So many of you are interested in complex tasks. Here are just one simple slide I put together on, what I like to think about, what are our brains good at, what are machines good at today. And, this is I'm not going to take you through all these, but machines are better at us than a lot of things, but in some things like object recognition or general scene understanding, or even things like walking well, machines are not as good as us. And our goal, I think of many of us, at least my lab, is to try to discover how the brain solves object recognition, and I mean that in an algorithmic sense, actually, potentially, that would mean that this would be commensurate with building machines. That's sort of a definition of actually saying that you understood the problem. And I also, more broadly, I think like Tommy and others think of this as a gateway problem, that understanding a complex problem like visual object recognition might give you more insight into how cortex works more broadly. So this is all to just set context here. So to back up a little bit, the goal of any science, if you will, is to sort of really big picture meta here, is to say, can you take measurements in some domain 1 and predict what's going to happen in domain 2? And this slide is partly, maybe I don't need to motivate for this computational audience, but more for my neuroscience and psychology colleagues, that predictive power is really what science is all about. This could be a future point in time, this could be the state of planets in time 1 and time 2, and we can do those kind of things with scientific understanding from fields like physics, but fields like neuroscience are still struggling to do this well. And for us, those interested in vision, this is a domain of all natural images, where each point is an image, so a point is some high-dimensional pixel space, and these might be mappings to what you might perceive in that image. And there could be various measures of perception here, and that's a very interesting domain, but that's the general problem, is you'd like to be able to take this and sort of predict what's going to pop into your mind. Now, the accuracy of this kind of predictive mapping is, again, the strength of any scientific field, and again, so the goal of accurate predictivity. I don't think I need to tell this group that, but in general, I need to tell neuroscientists that. And that's what ultimately underlies your ability to fix a system that's broken, to build something that's comparable to it, or to maybe even augment it. So just to translate this to the domain what I'm going to talk about today, again, the goal of accurate predictivity, for problems like visual recognition, this is face detection, which, as you know, is well, you may or may not know, but there's been a lot of progress in that recently. And so face detection was a pretty challenging problem for a while because of variation in the images, things I'll talk about in a moment. And so for that and other reasons, people started to go in and record neural activity. So now instead of just a domain of images, you have domains of neural activity where you can think of each of these dots as being, say, the spiking pattern of a population of neurons somewhere in the brain. We'll talk about those later. I just want to give you the big picture. And so the way that I think others that work in this way think about what we're trying to do here is we're trying to make predictive mappings between images and particular patterns of neural activity, again, population patterns of firing. And that's usually called encoding. And then we're trying to make algorithms that go from neural activity to accurately predict things like perceptual rapport, and that's called decoding. Okay, so that was sort of by way of big picture, because you're going to hear about those words encoding and decoding through the rest of what I'm going to tell you today. Here's what I hope to try to take you guys through today. First, to define a complex visual task. I'm going to talk, again, my lab works on core visual object recognition, so I'll try to define that for you, but I hope some of what I tell you today is inspiring for those interested in other aspects of vision. I'm going to then give a very brief overview of the brain regions that house the circuits that execute the brain's algorithm. That's a series of visual areas called the ventral visual stream. So those who haven't seen that, I'll take you through that. Then I'm going to discuss the central computational problem that you need to think about and how it's solved, or how it must be solved. Then I'll talk, as I mentioned, about decoding algorithms. So those are things like asking, have these neural populations effectively solve the problem? How does neural activity support that? I'm giving hints of the answers here. Is there any causal evidence in support of this? And I'll tell you a bit about that. And then in the last part, before we end, we'll probably break somewhere around here. The last part, I'll talk about encoding. What specific algorithm is at work along the ventral stream? And many people are interested in that, and I'll tell you what we've been doing in that and how we're taking a strategy that we think is making new progress on that. So I want to stop here and ask, since I've just been talking at you guys for a while, I want to get a sense of where you guys are in your thinking, and tell me which things. I don't want to just sort of talk to you blindly. If you're not stopping and asking me questions, then this will be a waste of time. It's good to educate you guys. So if people want to sort of tell me, do they feel like this is, if they'd like me to sort of skip through a lot of this, I mean, what's your guys' sense? Would people like to hear a lot of review of the ventral visual stream, a lot about object recognition, or would you like me to jump towards this? All right, how about, who wants a lot of review of the visual system? Or, half. Who's more interested in talking about decoding and encoding algorithms? All right, so let's sort of split. Okay, you guys feel like I'm talking, who's am I talking at too low of a level? Too high of a level? Okay, so I guess I'm doing okay. So again, I wanted this to be interactive, so please raise your hand, interrupt me. I will just, as Tommy sort of alluded to, I can just sort of machine gun fire this whole talk at you, but that's not what I think, this is really for you guys, not for me. So let's try to define a task domain, which I'll call core visual recognition. So again, I wanted to sort of point out that I'm not talking about all of vision. We're going to talk about something that I refer to loosely as object recognition. When I say that, I include things like face objects. I consider a face to be another object. So again, as I already mentioned, here's a scene. You might imagine, oh, yes, you quickly tell me there's a car, there's a person, there's a sign. How did you do that? Well, first of all, when I review the visual system for you, I'll show you that the ventral stream, it doesn't really analyze the whole scene at once. The anatomy suggests that the central 10 degrees is heavily represented in the part of the visual system that's solving object recognition, called the ventral visual stream. And so you can think of it as sort of heavily analyzing this sort of central 10 degrees. Now, 10 degrees for you guys, that's basically hold two hands up at arm's length. That's about 10 degrees. So that's to give you a feel for what we think your ventral stream is about doing. So for some of you, that might be about 10 degrees, depends where you are in the room. Hopefully it's sort of close. Okay, so that's a fixation point. So you might be fixating there, or you can think about 10 degrees. But when you explore the scene, when I put it up for you spontaneously without me instructing you, you make these rapid eye movements around the scene, called saccades. And you pause at these points here, which are called fixations. And the duration of the pause is about 200 to 500 milliseconds. So 200 milliseconds quick, 200 milliseconds quick. So you're making these sort of samples of the scene, and there's interesting science that maybe you guys or we can talk about is how you direct those gazes and the parts of the brain I'm not going to talk about today that controls that sampling. What I'm going to tell you is that given this kind of sampling, what this brings to your ventral stream engine, think about that central 10 degrees now, might look something like this. Okay, so those were snapshots at those fixation locations, each for about 200 milliseconds. And notice they were sort of in context, but I hope they were all drawn from that scene, so you had some context when you saw the scene, but I hope you noticed you can recognize one or more objects in each and every one of those. I'll do it for you again. You can quickly map it to some word in memory, maybe one or two words from each of those snapshots, even in only a couple hundred milliseconds. So this is what we call core recognition. And again, it's an operational definition of the problem that connects scene understanding to a more defined problem that we can go in and study quantitatively.
Jim?
Yes.
Is this like category recognition and not a specific exemplar?
So I'm being loose about that right now. So you mean distinguishing one type of car for another,
Or like,
but for now, think of it as basic level recognition, which would be the first word that pops into your head, which you might call a category. But there's of course different grains at which you might be able to do that. Was that car a Chevy or a I don't know, Honda? Maybe there's something that you can't do in those time durations. So there is a lot you can do, which I was sort of giving you intuition on, but of course there's limits to what you can do. But we're actually going to take advantage of those limits. So you could think of this as categorization, but it depends on exactly how you define that. I'd rather think of it as basic level recognition. All I'm trying to illustrate is that you have some power in that domain. What we have to do is then measure that power, and that's what we'll show you later. So I'm trying to set up how your brain tends to sample it, and part of the problem that we're going to study. So we're not going to do all scene understanding. We're just going to do what do you do in that snapshot.
But if it's a person, you can recognize a person.
You can, depending on the pixels, you can do some things. When you say if it's a person, it's under what conditions, it depends, right? So, yes, but I think I was trying to give you intuition that there's a lot of, at least words, single words that you can pull out of each of those images very quickly. Okay, did that answer the question you still want?
The only reason I asked that is that it seems like within category, it's like discrimination and the cross-category detection could be very different computational problems.
It could be. I think we and others tend to assume that they're grains of the same strategy, but I think that's an interesting point for discussion, especially when I'll show you the behavioral data, which shows that, of course, you do worse at discriminating among two very similar faces in that kind of time window than you would, say, a face and a car. So it doesn't mean the information content itself is an easier problem, even in the physics 3D sense. Right? So I think that's what you're alluding to is like, what extra mechanisms do we invoke the tighter the discrimination gets, which is usually called subordinate level discrimination as opposed to entry level. That's what you're after, right? Right. Okay, and I think that's an interesting problem. I'm only going to tell you behavioral abilities in this domain. And I'm calling it core because, again, I think that the things we understand from that will inform those more fine-grained things, but we'll have to demonstrate that for you at some point to convince you of that. I'm just telling you the strategy we're taking. Yes?
Have people done like Molly Potter RSVP stuff for subordinate categories as well? And do they have a sense of it, like kind of a quantitative drop-off or like a qualitative?
In terms of time of presentation drop-off? So we've done that, a lot of that, actually, that's a good, let me just show you, this is what I'm about to show you next. I call this ability core recognition. Again, that's just the operational definition of, say, central 10 degrees of vision, let's say 200 milliseconds, and you're asking about, so you could ask about within 200 milliseconds and then even beyond 200 milliseconds. And we plotted that out for tasks that I'll show you in a minute. And what happens is between 0 and 100, you rise up quickly from chance to a very high level. Then you have a little bit of an increase from 200 to 500, a very gradual increase. This is for single objects that the type I'll show you later. So of course it's going to depend on the task, but that's the general trend of it if you plot those data.
But does it interact with can you make kind of generalizations about subordinate? Is subordinate generally harder, or is it like some subordinates harder, or like does it depend on expertise, or do you have any?
Right. I mean, this is sort of getting to the space that we haven't gone into that both of you are asking about, which is if I plotted that curve for subordinate, would the later rise be steeper? And presumably it will be because there's more room to gain there, but what are the limits of that? Once we get beyond a couple hundred milliseconds, it's hard to control experimentally where your eye movements are. I should plot that for you, because that's sort of where we're going next. What we've been doing now is what do you do up to 200, and what I point out that you have this rapid rise up to about 100, and that's what we've been trying to understand. And you're already getting into like, well, what about beyond that? So hopefully I can convince you that maybe we have a sort of reasonable understanding of that first part, but the next part is sort of we call that sort of the next frontier. Okay. So that's the Molly Potter type video, right? You mentioned RSVP, so Molly Potter in the 70s show you can do videos like this, and you can usually quickly map one or more objects to memory. Now, you can attach a name to it. So this is just, again, this was known from the 70s that your vision system can do a lot, at least under some conditions, again, quite rapidly. I want to point out that it's pretty fast. I've already been saying that. It doesn't feel like you're putting a lot of effort into this, whatever that's worth. It doesn't feel like you're thinking about, long division or playing chess. And again, I think this is why early AI was sort of assuming that this problem could be solved very quickly. It's something our brains have been involved to do quite well. So we then sort of assumed that it was something easy to solve in a machine. I want to also point out that I didn't need to tell you to expect Star Wars characters. I didn't need to tell you to expect buildings in Italy, yet you were able to probably say, oh, there was a Leaning Tower of Pisa. I didn't have to strongly cue you intentionally. That doesn't mean there are no effects of attention. You do a lot without any pre-cueing. When I mean a lot, you're entertaining thousands of things that are quickly mapping to memory. Again, Leaning Tower of Pisa, Yoda. Who would have known that I would have shown you those things if you could quickly do that? So there's a lot you can do without these kinds of things. Again, not denying them, but there's a lot that already happens. And what's really impressive is it's very tolerant to variation, like his illumination on Yoda and so forth, so we'll talk about that again.
So are there some priming effects for under the 200 millisecond range? I mean, I would guess from what you're saying is that the priming effects would happen towards the end of the 200 milliseconds or afterwards.
It depends on the task conditions how much effects you're going to get. When you say priming, like if I cue you to Star Wars characters.
Right, before you start showing.
You're going to see one of the Star Wars characters. So it's going to depend on the specifics. You're going to see something in the center, to the right. These pre-cueing things are what I think of as what we call classic top-down attention, and those things do have effects. The magnitude of the impact on the behavioral performance depends a lot on the specifics, but they do have effects.
And that early?
Yeah, because you're pre-cued, so you can already see those effects very early. But what I want to point out is that those effects actually are sort of modulations on the larger effects we already talked about in terms of time. So we have been trying to focus on what happens even ignoring those effects in these neutral conditions where I'm just going to put things at you in unknown order, if you will. And because our argument is you already do a lot, and we'd like to understand that first as a baseline for understanding more complex rules. So again, I call that core object recognition. It's a subset of recognition, again, operationally defined. Okay, so let me give you guys now an overview of the brain regions that we think house the circuits that help execute that kind of task, and the pre-cueing things that were mentioned. So this is the ventral visual stream. Of course, this is the human brain. We all really would like to understand that brain. We work on this brain in non-human primates. Have you guys had any talks from non-human primate folks yet? No, no, not yet. Okay.
Yeah, no, we didn't.
Yes. Okay, well, maybe some. All right, well, anyway, what the non-human primate, part of the reason we and others like this model is it's a very good model of human abilities. Now, these are recent data from my lab because I used to just say that because it's showing low-level tasks. Here is the ability of the monkeys to discriminate objects under high degrees of invariance that I'll tell you about in a moment. These are the confusion matrices. So red means that it's highly confused, the blue means it's not very confused. Here's some object names on the bottom. These are just 24 objects. And all I want you to see here is that these patterns look very, very similar. If you did like a low-level pixel model or a V1-like model, we've done that. These patterns are very, very different than what you see here. So monkeys look a lot like humans in their confusions. These are monkeys that have been trained to discriminate these objects. It takes them a couple weeks to learn each object, but then they know many, many objects. So they need some learning, so they don't know how to respond to these objects initially. But I just want to point out, these are things that sort of feel intuitive to you. Camels confused with a dog, hiding by monkeys and by humans. Tanks are confused with trucks. This is not surprising. It's sort of in the shapes themselves, we think. But again, it's not like any algorithm does this. The point is that the monkey looks a lot like the human in these kind of core object recognition tasks. Yes.
What region is it?
Well, that's no region of brain. Those are behavioral data.
Oh, that's
Sorry, I wasn't clear about that. So a monkey is a good model for the human system, in part because its behavioral reports look a lot like the human reports.
So what's the behavioral report of identity?
So I guess I'll take you sort of jumped ahead, but the idea would be I put up an image, and then you have choices of which of those, here's 8 words. Was it a car, was it a dog, was it a plane? And it's sort of like the images I showed you, but one at a time. I'll show you those images a bit later. But that's the basic idea. From that, sometimes you'll say, oh, the camel was a dog. Occasionally. When I say you, that averaged over a lot of subjects. Those are the kind of errors that tend to occur.
Well, what does the monkey do?
The same thing, exactly the same thing.
Words?
They make up the car.
No words, you can do it different ways. They have picture match. There's like an image and then iconic pictures of the car and the dog. You think you could train with words, but we haven't done that as a location on the screen. It's not really reading, it's making an association, right? But the oddly interesting part is that the mapping between the images, which can be highly varied about a car, to which word he chooses. And, this isn't to say that monkeys know what cars are, and that humans know what they are, but at least in terms of these kind of visual tasks, they show the same patterns of errors once they're trained to do the kind of tasks. And we think all of us had to be trained to know what a car was at some point in our lives, too, to make a mapping between some feature basis to the word car. And we like to think that that's what we're teaching the monkeys, but this gets into deep things about the difference between humans and monkeys. All I'll tell you is operationally on these tasks, those are the data we have. Yes?
But then you teach them to classify actually.
We teach them to discriminate among objects, and they get awards if they can do that. And I could show you the training curve at some point, but again, it takes a couple weeks to learn every new set of 8 objects. You can learn like a new object roughly every day or 2 on average. So you can imagine we can increase their vocabulary very large. That's something we're trying to do.
It takes a day to learn a new object? That's probably not true for humans.
Don't know. This is deep and interesting, right? When did you have to learn the mapping to the word car, and how many training examples did it take?
No, I mean like novel objects.
What's a novel object?
Okay.
Right. I mean, people like to say that, but it's like we have, the way I think about it is a visual basis that all of us might have, might be built in by evolution, might be learned by statistics of the environment, some combination of both. But then you have to attach a label to it to actually do something to report on these kind of tasks. And we like to think that the monkeys and us share a common neural basis, but that they haven't yet attached labels that we have, and this is some evidence that we teach monkeys to attach labels, they make the same mistakes we have. So evidence in support of that idea. Again, there's some limits to how much a monkey's going to be like a human. Can you drive it? Dysfunctional goals are the thing that a monkey's not going to do, but in these kind of tasks, we don't see the differences yet between monkeys and humans.
With the task, there are 2 alternative sets.
For the humans, we've done a multiple ways. These data were actually 2 alternative choices for the humans, but they were not pre-queued. All these are interleaved. So again, it's important to intentional questions that came up there. There's an image, and then 2 choices. And you didn't even know what the 2 choices would be ahead of time. You just were ready for anything in a large world of, say, 20-some objects possibly appearing.
Do you have any idea why the monkey uses the watch and shorts on the slide?
That has to do with the specifics of the particular 3D objects we picked, probably. We could try to take it apart, but yes, at some level, but we haven't delved, these are actually pretty fresh data. We haven't delved into exactly why that would be. And we would think it's because, again, they're all basis for some reason, tends to put those shapes nearby each other, and then we're reading that. That would be our first hypothesis. But that's not answering your question, it's just shifting it to like, well, then why does the neural basis look like that? And that's the question that we would call the coding question that I'll talk about in the talk. Okay, more, yeah, in the back.
So, do you know what other species might also match the human performance in recognition?
We don't know. Again, this is the animal system we use in our lab. We haven't tested all possible species. I imagine, based on anatomy, that chimps would probably do as well as humans. Some kinds of monkeys would do less well. Rodents would certainly do much less well, just for acuity reasons. Because these are the exact same images for the monkeys as humans. So it's a really interesting question as to how that maps out. For us, we're just pointing out that in this domain, this is a model system almost quantitatively for describing behavior. So I'm sorry I can't give you any more than that, because this is all that we've done. Let me sort of then say, why the heck are we doing this monkeys anyway? I think you guys already know why not studying humans. Well, partly there's a lot of work on monkeys that tell us about the visual areas that are involved, called the ventral stream. We think these are the areas that are computing the representations, and I'll give you some brief evidence of that in a moment. You should know that neurons in these areas project to things like prefrontal, which is involved in decision action, these cortical areas up here, and around the bend to the neotemporal lobe, which is thought to be involved in things like memory. And so again, we think of these neural patterns or features, if you will, as the substrate or for the basis for what you would call higher level cognition, which is usually mid by these kindlets here. So you can see anatomically that makes sense. And we're not, of course, the first to point this out. I'm just orienting you guys. Because these are monkeys and not humans, though, we can study the neural activity at the level of neural spikes. And we think that's the most appropriate level of abstraction. But that's also an interesting point of discussion, how fine-grained these measurements have to be. But in monkeys, we can go and do that, and we can push the neurons around it. And I'll show you that, I hope, in a bit. And things that we can't do in humans. So we have just better tools to go in and dissect the system here than we do here. Okay, let's talk about visual recognition. This is a very simplistic view, but here's the ventral stream. As engineers, we like to break it down into these, let's call each of these as a visual area. Think of millions of neurons in each area. I'll show you that in a minute. Sorry, the pointer's down a bit. So what happens is when you get this image here, it produces a pattern on your retina, which is really a nicely processed photograph. It's rapidly conveyed up the visual system, and this implies a sort of feed-forward pipeline. It's not that simple, but just the average latencies here are 60 milliseconds to V1, and then on average about 100 milliseconds to start seeing responses in IP cortex. So that's partly those kind of data plus anatomical data that give you the layout of these areas. Now notice I have feed-forward connections with some unknown transform. I have feedback connections indicated with the dotted line. I have intercortical connections with this funny arrow. So it's complicated anatomy here, but this is the rough layout of it.
Jim, can I just get in your brain on pathways from V1 to IT, V2 to IT, and those kinds?
Yeah, the bypass pathway. Everything exists to some degree, and there aren't really great quantitative data on that. These data often come from making an injection here and looking at the density of connections in different areas. So I'm sure you go back and look at the studies in V1, there's some projection into V4, it's just a little weaker, and so forth. So quantifying that, though, is not something that's been done well yet. So I know that's not really an answer to you, Tom, but if you look at the dominant connections, this is the first order model. So please, I don't want you to guys walk away that's pipeline. The anatomy plus the latencies here, and the connectivity pattern between those, which I don't have a slide on, which layer of cortex. Remember the 6 layers of cortex. Maybe I should have a slide for that. These are not equal neurons. The 6 layers of cortex are all a little bit different. There's output neurons, there's ones that stay local, there's ones that project back. The anatomy of how they project back and forth is what helped establish the field, establish this hierarchical layout that you guys often hear about in the ventral stream and other cortical regions. But this is all a first-order model of how the system looks. Sure.
Yeah. Do you think the estimates on the latencies to be like overestimates due to experimental conditions, and that like in the natural world where we're constantly getting visual stimulus, this could be much quicker?
I guess it's possible. Again, those are average estimates. I have a latency plot I could pull up. There's a wide range, first of all, in each area. You're asking like if you change the precondition. I mean, if anything, these might be sort of, underestimates, because often they're measured from a blank screen to a sudden luminance transient, which is a pretty powerful pulse to the system. I mean, a new image on a blank screen is a better driver than like watching a movie where neurons tend to trickle along and you have a harder time estimating, making estimates. But I I don't know if that's, I don't think it's going to be much different. When you looked at when people estimate latency, and we've done some of this in our lab, where it is more of a continuous estimate, you do things like reverse correlation and look at the latency according to that, it's comparable to these numbers. I have a whole slide on studies of latency if you're interested, but I don't know if we should delve into that right now. But it's not going to be that much different than this. Okay? That would be my guess. I just wanted to point out that what we think about is like when you're watching the RSVP, of course, your homolog, your IT, your homolog of IT, and your other visual areas in your brain that are homologous with the monkey brain are clicking along, populations of neurons firing. Just to give you ballpark, about 10% of neurons in each area are on average active for each image that's shown from a set of natural images. Okay, so they're all firing in there in response to the images, and of course we can go in and measure those kind of things. And what my lab tends to think of, and I think lots of labs, think of each area as containing a new population representation. And the reason we and others think that way is that we know that there's a complete retinotopic map in the retina, of course, in LGN and the thalamus. I'm sorry, this is dead. In LGN and the thalamus, in V1, in V2, and V4. In IT, things get murkier. I'll show you some data on that. It's maybe more than one, probably more than one area, but it's almost getting less organized about the spatial layout and more organized about things like categories, things like bases. So I'll show you some of that data later if we have time. Everybody know what a retinotopic map is? Okay? Don't be shy if you want to. Okay. And again, I don't want to go too slow. Okay. So here's a drawing of the kind of rough size of those areas. Now, this is a little more drawn to scale in the monkey so you can get a sense of things. Here again on the latency, each area is sized to represent the approximate number of neurons, and the color bars indicate sort of roughly the central 10 degrees, right? Not all of V1 is dedicated to the central 10 degrees, so the color in V1, I try to scale to estimate what portion of V1 is covering 10 degrees. As you can see, IP is sort of heavily, almost all filled by the 10 degrees. I want to point this out because you guys are, if you hear about other visual areas, you might hear about people studying MT. Look, there's MT, little tiny dot. So there are lots of people studying these kind of things, and you probably know a lot about those studies, but there's a huge chunk of brain devoted to vision that you sort of see here, again, called the ventral stream, especially the central 10 degrees. So again, I've indicated IP here as three areas, eight IP, which is anterior, central, and posterior. And people parse in different ways, but that's the way we just like to talk about it.
This is more or less in scale?
Yeah, well, I mean, scale, these are scaled relative to each other. So in terms, you can see, I don't know if you can read that, 190 million neurons here, there's 36 million there, there's 10 million there.
Is MT as small?
Yeah, MT is scaled relative to these other areas. Right? And Tony Momsen likes it, it's called a pimple on the floor of the SDX. And Tony Momsen is a big studier of MT. He'll be also the first to point out that a bunch, if you look at the number of researchers, huge here, very little study up here. This is a difficult area for, it's only now that people are starting to do this in the last, say, 10 years. Yes?
I was wondering where that 10% number came from, that you said 10% of neurons were active.
Oh, that is some studies, partly from my lab, partly from labs like Jack DeLance. We don't have time to tell you, but Nicole Russ recorded a bunch of neurons, shows a bunch of images, and then just counts. That number is roughly how you count neurons active is driven to half its maximal firing rate observed among a bunch of images.
Do you have to worry about sampling biases?
Yeah, you do, but when you look at the two photon data that people have more recently, it doesn't seem like there's a lot of dark neurons, if you will, so you might worry that we're sort of overestimating, but so there's some worry there, but it doesn't look like it's a big worry. And Nicole, in those studies, she would take any neuron as spiking at all, but you might worry there are neurons that just never spike and then show up occasionally, but at some point, we can estimate that they're not spiking at all, become insignificant, at least with respect to the domain of the task, and then we're going to fire for anything months to do the task. We don't care about that like that. So it might almost think of the glia, if you will.
I have a question about the latency. It's not that I don't trust the number, but it seems like our human, we can recognize the gist of the scene in just like 25 milliseconds, but the time when that is probably just still at V1 or even not reached.
Okay, let's be clear. You can't report 25 milliseconds correctly, but you can deal with a 25 millisecond input. So there's a difference there, right? So this is the input coming in, it can be short, but then you're not going to give me a response, or you probably not, so at least 300 milliseconds. For a monkey, maybe around 200 milliseconds to actually press a button and make an execution. So there's a time lag to the behavior. But you're right, you can put smaller pulses of data in that are going to take a little while to propagate through the system. So I think when you're thinking of 25 milliseconds, that's the image time, not the reaction time. That answer your question. So we think a reaction time is pinged by, depending on the task, You know, it might be longer or shorter. If they're cats that are somehow leaning on these neurons, you would expect them to be shorter than what is required of these neurons. Whether that could be measured behaviorally, no, I don't know the examples of showing that, but that would be the prediction of that.
Is it just a lag or a buffer time, or is it process time? Can we process?
No, but, well, okay, what I can tell you, one thing that I think is interesting to point out, especially for this audience, the time between here and here given axonal conduction delays should be about 1 millisecond. So these latencies have to do more with cortical integration than do with like just transport time. Okay, so the speed of the action potentials from here to here is, and given their distance, suggests that there should be a 1 millisecond difference if you just sort of went all the way up here. Remember that synapses, axons come here, synapses have to release, neurons fire at layer 4, they transmit to layer 3, 2, 3, those transmit to layer 5, 6. All that sort of churning is happening in here, and then you see these latencies up here. So but also don't think of this as bang, bang, bang. Think of this as like this rolling wave of activity, right? So it's very overlapping. There's large variance in latencies. These are sort of the average, the median latencies. You're going to have that slide. Maybe we should come back to
But
it.
can we recognize sequential images, or we need, or there's a buffer that will fill up, and we can't just show sequential images?
I don't know if I want to use the word buffer. All I can say is that these are the averages to a bunch of images. So maybe we can sideline that question in the interest of time for a moment. If you want to come back to it. It's something I should pull up a latency slide during break if people are interested. Let me just quickly say the reason we're in this part of this brain is that if you lesion this part of the brain, you get deficits in monkeys, for instance, being able to discriminate this object from that object. It was studies like that led people to think that IEP cortex or the ventral stream in general is important to what they call what, and that, for instance, the dorsal stream, this part of parietal cortex is more involved in location information. This is very oversimplified, but that's part of the reason that motivates the ventral stream to be involved in this task. So let me give you the brief tour of what's going on here. So here's the retina. So I'll show you one slide on retina. Yes, retina has basically nice center surround pixel detectors. They respond to this is a pixel, if you will. This is these they're tiny little receptive fields. Depending where they are, they can vary in sizes over a wide range, but they tend to be about, let's say, about a quarter degree, or actually a little bit less than that. I'm sorry, that's depending where they are, they can vary from very small to pretty large, but they respond to increases when light comes on, and that drives the neuron more in the center, and if you put light on the surround, this is an on-center or off-surround, then you suppress the spiking response. So what I wanted to tell you is on and off-center ganglion cells, they're pretty linear, there's nice gain control mechanisms, and there's a lot of work in the retina, beautiful work in the retina, but from the point of view of an image, they're just sampling tiny, tiny little samples, almost pixels, but well-processed pixels. Maybe like your camera does a really good job on your phone of actually kind of producing a nice image out of that. The retina has little tiny center surround cells. If you go up into like B1, this is old work from Buble and Wiesel, but you guys should all know about it if you don't already. Cells have what's called orientation selectivity. They're called simple cells. And what that means is they respond to, say, a particular line orientation that can be modeled quite well as linear filters that have plus and negative regions. I know you guys can't all see that there, but those are built up by the LGN cells converging in the right way, and that gives rise to a selectivity where it likes this orientation and likes, they can see responds there. That's what I mean by likes. You change the orientation, you get less spikes. That's called orientation selectivity. And then there are other cells in B1 called complex cells where they have selectivity, they like this orientation and not that one, you can see that here, but they also have tolerance, in this case, of position. And this elemental operation inspires many important models in the visual system. It still inspires them that you have this buildup of both selectivity, as sort of described there, and then a little bit of tolerance, in this case, at least to position, that described there. And that sort of central idea, again, inspires what I think Tommy's group and others have built into many models of the visual system, based on these V1 kind of observations. Yes?
Are there differences between the retinal ganglion cells and like LGN? They're both center surround.
Those are generally very, very similar. People
What
work
gun
on
do you have that? That seems weird, right?
Yeah. Why would it just be a relay? Like, what are the processes to go on? Again, evolution may do things that we wish that they should undo in some way, but when people measure it, those cells look very much like functionally like retinal ganglion cells. And so it doesn't mean that there's no other thing going on in the thalamus there, but just functionally, you just did linear filter maps of them, and they look very similar to the maps I showed you for the retina.
Like same size on and off. Like
They have the same on and off heights, and they're very similar size. It's not our active area of expertise, but when I read about those things, that's how they're portrayed. I don't know if you guys are having anyone from the LGN community or anyone in the room that's Gabriel. I see, is that Gabriel back there?
Yeah, no.
Do you want to say anything more about that I maybe
No, I'm saying that there's nobody talking about.
Okay.
But there is lots of times between them.
I guess maybe the temporal kernels are a little bit different. There's certainly there's nice studies on, because there's feedback to LGN, but not to V1, or not to retina, so they may be interested in controls involving feedback there. That's something like Meadows group have looked at a number of years ago. But functionally, most of the visual community thinks that it's very similar, almost relay-like. I must say that every time gets upset because there's more there. But just from a filter mapping point of view, you can explain them pretty well with center surround filters. Yes?
So are these like cells from V1 or?
This is V1, so that's primary visual cortex right there.
Right, so do you actually see any orientation variant, tolerance to orientation as well in V1 by any chance?
Oh, well, okay, so the tuning width of these cells is not, they have a bandwidth to their tuning, right? So they like a preferred orientation where they have a range, of course, it's going to roll off as this model would predict, right? As you slightly tip that thing, you're going to get slightly less response.
Oh, I'm sorry, I was talking about complex cells. So do you see any?
Complex cells have a similar bandwidth orientation selectivity, but then have some ability to tolerate the position, and these can be fit pretty well with a linear filter, and these cannot.
But they don't have orientation tolerance as well.
Well, again, depends what you mean by that word. So there's some range in which they will respond. So maybe we should define tolerance a little more clearly. This is how people have usually used the word. That means that it's going to respond similarly across a change like this. I can tell you a whole story on how I think about tolerance, but I think maybe I should leave that for a little bit later. If you're still confused about that, ask me after a few more slides. I wanted to say, of course, I'm doing nobody justice with these. I can't review the whole visual system, but I'm trying to give you a feel for what's known about these areas. There's been more recent work, I'm skipping over this, on V2 and how it differs from V1. There's some beautiful work from Tony Options Lab suggesting that V1, basically V2 is doing combinations of V1 cells, products of V1 cells, and that is work that I don't have a slide for you today, but those seem to be consistent with the ideas of many models that you're just going to build up selectivity, but the particular type of selectivity of which V1 cells you cross together is still, I would say, a mystery from those kind of data, but there's evidence that suggests that you just take, you repeat this idea of what you saw, V1 being a combination of LGN cells, and then you have some buildup of tolerance in the complex cells, and then take the output of those complex cells and build products of them, if you will, another level of selectivity, to templates on those complex cells. That idea is consistent with the kind of data that they have. For a long time, it was hard for people to even distinguish between V1 and V2 with just using those kind of simple bar stimuli like I showed you. Like everything kind of just looked like a complex cell. So you had to use natural images and to source this out. And I'm sorry I don't have a slide for that, but I want to tell you that there's some advance there. I wanted to give you a history of the field, though, a little bit. If you go in and record in these higher areas, V4 and up into IT, I would be remiss in not showing you kind of slides like this. People for a long time would go in and record these areas, and they would say, well, I think the cells might like, hyperbolic gratings. This is, I think, Jeff Gallant's work. So they would show, here's an example of a cell. The red indicates high firing. This is single cell. This is another cell. The blue indicates low firing. And you say, well, look at like these patterns and not so much these, and it's like this one. And so you can find a long history of like, here's some cells that go zip and some that go zap under a particular stimulus set, and maybe theoretically motivated by some reason. But it was really hard to make sense about what these cells are doing. So it's viewed as a collection of interesting data, but without a sort of unifying theory to kind of predict it all well. Similarly, at Connor's lab, they showed beautiful work on measuring cell responses to curvature. They would use stimuli like this. I'm sorry, I won't have time to take you through this in detail. But, you could think of a simple object like this one as having curves relative to some object center, different amounts of curvature, and different segments that are curved, and different orientations of those curves. And they devised a basis for those kind of things to describe these shapes in a curvature basis. And they could then then go ahead and do, so they had this hypothesis that V1, V4 neurons are tuned in this, or sort of mid-level visual system, the V4 neurons are tuned in this basis. So it was just an idea, and then they would go and say, well, let's now test that idea with a bunch of, again, simple shapes. You can see the responses here of a bunch of cells. Black indicates, sorry, this is one cell. Black indicates high response, gray indicates, or white or gray indicates low response. You can see, well, like that thing, like that thing, like that thing, like that thing. You can see it like some of these weird kind of potato-y looking shapes where they're varying curvature. And from that, then they could build models where they would describe each of these shapes in a Bayesian set, that's the curve relative to the center of the object. Again, details that I'm not going to have time to take you through, but they could explain, half the variance of V4 cells with that kind of model. So this is just to give you a sense of the work that has been done in these areas. These are not things that are easily computable on actual images, which I think really limits them as predictive models, but it was sort of a kind of descriptor of what's going on at the level of the visual system for, at the intermediate level of the visual system for some types of stimuli. So this is just, again, a little bit of the history of the field. The simple way to take this is they showed you for a bunch of images. Here are some responses of those. We can't yet fully predict those well from natural images, but they have some progress in a limited domain of stimuli. And people that have looked, so those are kind of things, if you go look at the before literature, what you'll find, those kind of studies are the dominant physiology studies. If you go up into IT cortex, what you'll find then is studies that look, so I'm going to skip through, this is the anatomy about the central 10 degrees, we'll skip through that. Here's sort of now the history of IT cortex. So people that started studying IT come from a kind of different tradition than those that were studying V1 or the LGN. They come from more of a psychology tradition, and Charlie Gross's lab was sort of a big sort of beginner of this field, and at first a lot of this work was dismissed, but I think later turned out to be very important. And this is some of the early work from Gross's lab. You can see this in 1972. And they were in this mode of Hubel and Wiesel had just discovered these simple cells in V1, and they like to respond to oriented edges. And so here they are recording in these mysterious areas of cortex that you saw deep in the ventral stream called IT. And they would start showing stimuli like this. So they were trying to drive the neurons with various set-out stimuli to figure out what makes the neuron go. He was looking for the trigger features of the neuron. And, I'd like to show this quote. So the use of these stimuli, you can see them up here, was begun one day when having failed to drive a unit, a neuron. Drive, of course, means produce lots of spikes. I'm sorry I should have said that. With any light stimulus, we waved a hand at the stimulus screen. So they were trying the lines, and people went and said, well, and they do this. This is what they said. Waved our hand at the stimulus screen, and elicited a very vigorous response from this previously unresponsive neuron. So that's sort of anecdotal, but it sort of gives you a sense of what the field was doing, and this is what Charlie wrote. We then spent the next 12 hours. This is an anesthetized monkey. The eyes are propped open, and they're showing stimuli at it. Spent the next 12 hours testing various paper cutouts in an attempt to find a trigger feature for this unit, as if there's one thing that it really wants. When the entire stimulus sets were ranked according to the length of the response they produced, we could not find a simple physical dimension that correlated with this rank order. However, the rank order of adequate stimuli did correlate with similarity for us to the shadow of a monkey hand. So these are the kind of stimuli they produced. This is high responses or low responses from these kind of things to that. It looks like it's a hand neuron. So from that launched the whole idea that the trigger features of the neurons are kind of full objects, things you might attach words to like hands. And there is selectivity of these neurons. I'm telling you this is the history of the field. Whether these are strictly hand units or not has been still effectively an ongoing debate. From Charlie's lab, Bob Desimo's group showed similar things, I think, much more quantitatively. Here you can see these are post-stimulus time histograms. These are basically sums of lots of presentations of these images. Just look at these blips mean lots of spikes coming out. Those are, again, post-stimulus time histograms. There's the stimulus on here, here, here. In each of these cases, these are the visual stimuli. You see this likes these kind of hands, doesn't like the space, doesn't like that box. So there were these games being played, like, let's figure out what these neurons do, and we have a limited set of stimuli, and this is the stuff we try. Tanaka's group did similar things. Oh, here's a neuron that seems to like a face, even a reduced face. Take away this mouth line, it goes away. Take away these little eye dots, goes away. They bring it back over here, change the contrast, well, it kind of went away. So you'll find a lot of papers through the 90s that then look at these, they take this approach. Let's figure out what these cells are doing, especially from Tanaka's group. And they try to do things like reduce the stimuli. What features of the IT neurons tuned to? They take an image, a bunch of objects, they dangle in front of the monkey and figure out, oh, the neuron seems to respond better to this object here. Then they try it on a computer screen, try to reduce it to something minimal. And here's that it's saying, still responding, still responding, still responding, still responding. Now, when I do this, no more responding. This is how they present the work. And from doing that kind of thing, they would say, well, here are the initial objects, we reduce it to this. So one thing I think you can take from this is that they were usually able to reduce the stimuli to something that you would call more intermediate than the full objects they started with and still maintain a reasonably good response out of the neuron. So that's already a clue that these neurons are not, kind of think of them as object detectors and that they need to cat. Like this one, maybe some textures oriented in some funny ways enough to drive as well as cat image. These were just what they started with. Remember, they're trolling around in a super high dimensional space so that, it's just I'm not telling you this is like our understanding of IT, this is the history of what people have done, okay? You guys all follow all this so far? Again, I'm not, this is what you do when you don't know what to do, so I'm not trying to badmouth it, I'm just giving you the history of the field. And from this thing, then actually did things like compared from V2 up to post-ant. So here's the brain, here's V2 way back here, V4, and then IT coming down here. You can see the recording locations of those dots. And these are the kind of features that they would call the minimal to drive the neurons. And one thing they wanted you to eyeball this and say, these look somehow more complex than those. Yeah, this is, you have to, how are they judging that? And they're just giving you a plot for you and then hoping that you will take the story of complexity. The receptive fields, they and others have shown get bigger along the ventral stream. So you record here, the receptive fields here are shown as squares. These little squares, and you can see they get bigger. This is a 10-degree field. Remember I said about the central 10 degrees. You see a lot of sort of 10-degree size fields here, sometimes bigger, sometimes a lot smaller that my lab and others have found, but they don't show those here. But you could say on average about 10 degrees. So the receptive field sizes are getting bigger down the ventral stream, something that you've probably heard about. These are the primary data. From those kind of data and others that I won't show you came this very simple conceptual model that we still carry around that basically goes like this, that neurons and IT, they have some receptive field indicated with the dotted line, so the portion of the visual field. They respond to some object, well, again, many objects or many sort of shape features, which we don't quite understand. Here I'll just symbolize the letter A. And they respond to that even if you kind of shift it around inside that field. So remember that complex cell in V1 that lights that bar? It's the same thing, except the field's bigger and the shape more complex. So it's now an A being shifted around inside this bigger receptive field, and it can change the size and it will still respond well. But then if you switch it to another set of features, it didn't really respond well. So here I indicate that with V. So this was the sort of qualitative, conceptual story about what single IT neurons did. Of course, the detail is still not understood, but it's sort of you see an extension of, if you will, a visual to a more complicated shape space and bigger receptive field.
Are these also invariant to rotation, or is that?
To some degree, and Nico Logostidis, with work with Tommy, sort of has a really cool study on that in the 90s, so I was going to mention that in a moment. There's some degree of tolerance to rotation, but it's more difficult, it hasn't been fully fleshed out quantitatively. It's more difficult to give you a clean answer on that, but you can find neurons that will respond to, you say, an object even across large changes in rotation. But then you'll find other neurons that are tuned to various particular views of that object. And so you kind of have both types, but of course you can take view tuned neurons and pull them together to build up fully invariant neurons. So there's some, just like here, this isn't the whole visual field, there's not the whole rotation field is the way to think about these neurons. It's just within some range of rotations, they have some, they're still responding better to their preferred object than to the least preferred object. So that's what I mean by tolerance. You maintain rank order selectivity across some range, in that case of pose, in this case of position. You could also imagine that kind of curve scale. So the most important thing computationally is that the response to B doesn't rise above the response to A over some range here. And that can be for position or for pose or for anything. So that's what I mean by tolerance. So the question that came up earlier, if you have neurons that can do that like object A better than all the other objects over some range, that's actually a really, really good basis for discriminating with objects and maintaining information about things like position and scale. It's just separated, jointly coded two latent variables, one about position, one about identity, or one about pose and the other about identity. So if you think about this, they're individual neurons, but if you think about a population of them, that's the key single unit property you would want if you're going to separate out these types of latent variables from each other. I'll show that in the next slide. Okay, is everybody with me so far on my brief whirlwind tour of the ventral stream? Okay, so this question was just asked. IT has some tolerance to viewing context, cues that define the shape. I didn't show you that. I mean, exactly how to define the shape boundaries. Interesting work on that, the retinal position and size. I didn't show you the data, I just described it. This was just discussed in the Logothetis studies, the Booth studies, that both suggest some tolerance to view, the illumination to 3D objects, and some tolerance to this, some ability to tolerate visual clutter. Again, all these things are sort of partway. They're not perfect, but there's some ability to deal with these things. I also want to then say, remember, we're dealing with a piece of tissue, right? So what I was describing so far is this, there's a bunch of neurons, and with the sample amount, I'll tell you about them individually. The neurons are not in a vacuum in a big mixed-up bowl. They actually have some spatial organization in them. So neurons that like nearby things tend to live nearby. Just like in B1, neurons that like the same position of the visual field tend to live nearby. That's a retinotopic map. Or like similar orientation, live nearby. That's an orientation map. In IT, there's some sort of spatial structure that's on the scale of around 500 microns to a millimeter. And this is Tanaka's group is the one who first pushed this idea, and they went in and when they reduced their features, they would show that nearby neurons tended to have similar features. And they kind of drew it like this in the hope that this is just a visual model of what they thought was going on from their data. I think the data, I think Gabriel Kryman and with Chao Hung working in my lab actually showed a very similar thing later. So I think there's some truth to the idea that nearby neurons in IT respond to similar things. There's some structure, again, at the 500 to 1 millimeter scale. This is optical imaging data of IT. I don't have time to tell you about optical imaging, but just think of these patches as being the response area to these particular stimuli. Don't worry about their crazy cats. All I want you to see is that their blobs here are on the order of about 500 microns to a millimeter. So you get these like regions of activation, and they're different depending which stimuli they show, but you can see they're getting sort of chunks of tissue activated. Okay? I don't want you to think every neuron in there does exactly the same thing, but they're just more similar to each other than say, these neurons are more similar than say a neuron here and a neuron there, which are almost, when you go far enough apart, are very dissimilar when tested with a large number of objects. And that's the kind of data that I think that Gabriela showed that that those things, that there is there's spatial structure at the scale. That's how we can quantify that. Then I want to point out that more recently, Doris Sal's lab, working with Marge Livingston and Vivek Frymwald, showed this spatial structure even sort of slightly larger scale. I already alluded to this with faces, but you go up to IT Cortex here, they were doing image contrast motivated by Nancy Canwisher's group. They were saying, hey, there's regions in the human that respond more to images of faces than to non-face objects. And they show that there are these sort of regions that they named here. These are their names. I don't need to take you through them. But there are regions of tissue that respond more to these than these. And now the size of these regions depends a lot on MR things, how you threshold this. These are MR data, so functional magnetic resonance imaging. And this is all of IT. It looks a little weird to you, but IT is basically from here to here because of the way they rotate and inflated this monkey brain here. But there are these structures in there, and Doris and later showed that if you go in and record from at least the middle patch here and later from the anterior patch that she and Vivek showed, that the neurons, a lot of them, as you might expect from the MR, tend to respond better to images of faces than to other things. So this is a more modern-day version of what Charlie Gross and Bob Desmond had originally said. Their neurons respond well to faces, but here they're spatially clumped at scales on the order I got up there 2 to 6 millimeters. There's some clumping of neurons that at least like these kind of face images better than these other images. So people tend to then call these things face neurons. I think that's too strong, but operationally, if you test them with a bunch of images of these kind of things, these are the data you get. I'm sure I didn't take you through this. The red means high response, this means low response. You can see almost all of these neurons tend to respond better to images of faces than to other things. That's what's on the right-hand column there. Okay, the point is that there's structure at slightly larger scales, maybe for classes like things like faces. This is just to show you from my lab, get just a more complete picture of that rather than MR. This is Elias's work in my lab. You go in and record the IT cortex, and you just, each of these dots is a recording location. He's measuring the response to, again, a bunch of images of faces versus non-face objects, not just 2 images, there's a whole battery of images there. And then the color indicates the degree of selectivity of the preference for faces over non-face objects. And what you can see, so this is more of an unbiased sample of what's going on in the ventral stream, and what you see is, look, kind of some red clumping here and sort of here and here. These turn out to line up very well with those MR patches that we measured in the same animal. This is called the posterior patch, the middle patch, and the anterior patch. So you can see that there is some, again, clumping at this larger scale for these kinds of object contrasts. Yes?
I have a question about the point made on the previous slide. So there are a lot of dimensions along which things could be similar and neurons may respond to similar things, but the brain is embedded in some like 3-dimensional blobs. So you won't be able to replicate all spatial similarity all similarity, spatially.
Yeah,
So,
you gotta put a whole bunch of objects on a 2D map, is what you're saying, yeah.
so, yeah, what challenges that pose to like?
I think this was a great point of discussion. I'm giving you a review of the literature because I'm trying to be fair to what's out there. I mean, I have my own biases on these things, and there's a huge debate about this. I should say more recent work from Bevan Conway's lab, if you just do color, you sense those that are sensitive to color versus non-color, they get little patches that are here, here, and here. So to me, that looks like repeating random topic structure in a more complex shape space and not modules for doing faces and color. But the folks that are doing that work like the idea of a module, and that is actually, that connects well to our own ways of thinking about understanding. But my guess is that these are more of a continuum on a complex shape mapping, that when you do, there is some clumping, there's some natural grouping of the cells, and then when you do these things, you'll get what tend to look like clumps. And that doesn't mean that doesn't discount the data, it's just sort of how strongly you want to think of these as being walled off in terms of processing that. And the deep questions for this field are like, do those neurons only support your ability to discriminate faces, or do they also support your ability to discriminate a clock from another clock, which has round features, but it's not, no human would ever call either of those a face. So that's the crux kind of question that's still debated, and we're getting close to being able to answer that question. I think that's the bottom line. These are the data. The data show if you in-mark contrast with this, you get these flaws. The inference from that is where the debate is in the field. And I think you're kind of coming on one side of your questioning, I think, fairly, is how do we interpret this? Right? I'm telling you the data. Interpretation is the fun discussion on the edge of science.
Have there ever been neurons that not only are sensitive to faces, but have been identified with, like, individuals? That would be, like, sensitive to an individual monkey or a person?
I'm thinking about face categories. So the logothesia study that I briefly mentioned, but I didn't show you, and the Booth study that I mentioned, both had trained monkeys to discriminate among 3D objects or to know a bunch of 3D objects, and they had different kinds of training procedures. Then they had neurons that would be tuned inside the space of the things they had been trained to discriminate among. So you could call it individuation within the neural population that they could find. So I think the answer is, like, if you train animals to discriminate, then you kind of find a neuron somewhere in IT that shows sensitivity to the thing that the animal had learned. Now the debate becomes, well, how much of that was there in existence and how much of that was trained in for that task? There's various studies that sort of speak to that, but I think the answer is, of course, somewhere in the middle. Some of that was already existing, some of that was set by evolution, some of that by development on other natural images to the question of novel objects of that's here. And then some of it during the training period in which you enforced this is the discrimination habit. Yeah. That's my guess, but sorting that out is still, nobody's done that well. It's difficult. You have baby monkeys and control all the visual statistics to really dig into those questions. I think that's where your question is sort of leading. Because what we know as individuals sort of depends on our own history. Yeah. Right? I mean, I don't think we find, that being said, the monkey's retina can discriminate you from someone else, an image of you, one image of you from some image to somebody else, because the information is there on the pixels. So, but how that maps to the animal's perception, which is a deeper question, is probably going to require some training. I don't know if did I answer your question, or should I try again?
No, yeah, I guess.
Okay. Let's think about it some more. Again, all these are super cool, interesting questions. I'm trying to just give you guys a review of sort of what the field's gone through and where it stands right now. But I want to then kind of, since this is a computational audience, I want to come back to object recognition. So I went through sort of the ventral stream, but I want to talk more about the problem of object recognition that we started with, and what's the problem that needs to be solved, and then we'll take a break and hopefully get to the last slide. So this should be pretty quick for this audience. The problem of object recognition, I sort of already alluded to this, even if you take away color, there's many possible objects. On the order for base level object recognition, on the orders of thousands of estimated objects you can discriminate among. These are some that we use in the lab. These are 3D models just to give you some sense of objects. I don't have to put that on object, so there's many of them. So you need to deal with that problem. But you need to deal with that problem that would be called selectivity among these objects in the context of being able to tolerate the fact that any one of those objects, like this car, can produce an essentially infinite number of images on your retina due to the fact that you don't just see the car just like this all the time. You can see the car in different positions, poses, or different illuminations. You might even, to the point of categorization, want to say, well, look, these are all cars, so this is exemplar class variation. And of course, a car can appear in backgrounds, many different backgrounds. You can have occluding things. You still want to call a car. So there's a lot of needing to deal with those kind of image changes. But all of these humans can kind of quickly say, that's a car. And I say can do that. The degree to which we do that is an empirical question that we have to measure, and we have measured it. I'll show you in a moment. But this ability to be able to say, this is a car, and not one of those other 600 objects, or, well, I actually showed you 50 some on the last slide. One of those other 50 objects back here is not, is called tolerance. So to say, oh, that's, all these are cars, and they're not any of those other things across all this, that ability is called tolerance, or sometimes called invariance. I don't like the word invariance because it implies perfection, and we certainly don't have that in our own visual systems, but that's the colloquial term, invariance, or the ability. Okay, so that sets up the problem space. Why is this hard? Again, I like this geometric interpretation. Some people in my lab don't, but I'll just give it to you. You guys can see what you think. So this is the, think about neurons as living in a state space. Okay, so each axis is the activity of a neuron, and there could be many neurons. Of course, in the retina, there's about 1 million random ganglion cells. It's a 1 million-dimensional space, and when you see this image, so this is an image of an object. So it's a projection, a 2D projection of one object, a source that happens to be a person we'll call Joe. Then because it's an image, it gives rise to a pattern on this 1 million neurons, which gives us one point in this 1 million-dimensional space. When this, the interesting thing is when this object undergoes a transformation of one other latent variable, in this case pose, it's not just jumping all around, it's a continuous latent variable, pose, but it's producing different images that then are moved to different points in space. You can see it's not linear in the pixel space, it's going to be some complex curve. So here's this other image of Joe. Those are all images of Joe. And then here's the other latent variable, the other axis of pose, and that's a two pose axes. Now he shifts up that way, now he's looking upward. But these are still all Joe. And you could imagine, not just the 3 degrees of pose, but you could imagine the 2 degrees of position, or the third of size as part of position. You just even think about those kind of latent variables. And of course, there are more, as I showed you in the last slide, but it gives you the sense that there's this continuous sheet of points that Joe could give rise to in this space here. And that continuous but highly curved up sheet, I call that Joe's identity manifold. And any point on that sheet should be categorized as Joe. And Joe is the source of that. Joe is what gave rise to that. If your neural activity was here, your best report right now is that's Joe, and not Sam or Steve or Jill or whoever. But it's this very thin sheet of points that's highly curved up in space. And so now we have to come back to well, why does this matter? Well, somebody downstream, something, not a homunculus, but some mechanism downstream, looking at the population neuron, has to make a decision about, when I put it in front of the monitor and say, is this Joe or not Joe, test it behaviorally, it has to make a decision. Is this Joe or not Joe, or Joe is to be all the other distractor objects that it has to deal with. In this case, it's called Sam. And each of them, of course, has a manifold of points that it could give rise to. And what I'm showing you here is a simple idea of separating hyperplane. There could be other simple mechanisms, but they're simple with respect to this population space. We like simple mechanisms because we think of them as implementable, what downstream neurons could do. These are linear classifiers for us, weighted sums of population activity with a threshold. That's why we like this. I know machine learning people like classifiers too, but we think of this in a simpler sense. This is a biologically plausible thing. It's a thing engineers know how to do. Many of you guys already know how to do. It probably maps to something that downstream neurons do. Earl Miller and Tommy showed, for instance, in prefrontal cortex, there are neurons that seem to be able to read off of IT to do categorization tasks. So there's evidence that downstream neurons reading from the ventral stream could use these kind of mechanisms to solve these kind of tasks. Okay, so again, we don't think of this as us going in and trying to extract information from the brain. We think of it as asking if there was a mechanism in the brain, here's the kind of mechanisms that we think are within its toolbox, and that's why we use these as sort of what are the downstream neurons that are viewing this, what could they easily do? So I wanted to say it's not just linear separability, which needs to be done here for this to solve. We should be able to find that plane with a relatively low number of training examples, and we'll talk about that later. But that's sort of the idea of what you want, is that usually with a few training examples, you have a simple decoder that can tell you, you can now do all of the object manifolds. So a few training examples then allows you to always separate Joe from object. That's the ideal of what you want for a task like this. And so we think of this as shown here
If it can support that, then we call it explicit. It's still a population representation, but it's explicit because there's only one little step of training needed with a simple decoder to then extract the information. So that's when I say explicit, that's what I mean.
So are the transformations that lead to the separation supposed to be static?
In this case, these would just be different static images of Joe. So,
sorry, I mean, presumably you're going to see Joe under all sorts of different eliminations, inclusion conditions, and stuff. Like, it's the idea that the downstream, neurons throughout the visual hierarchy, adjust themselves to maintain the separability, or is it?
Well, so you're asking sort of how, I'm just giving you the sense of, this is what a good representation might look like. You're asking how you actually build it and get to be that way.
Yeah.
I'm saying this is what you need to support this task on the core recognition domain. Here's an image, flash, is it Joe or Sam? You need to pass it through some decision to press the right button if you're going to do this well. You're going to need a representation that looks like this somewhere. This is my special claim. That's all I'm saying. I didn't tell you how to do this. This is just to give you a sense of what I mean by good. It also leads to our tests of what we think is good. It's by using population tools to ask, is the information explicit, and this is what I mean by it. That's all I'm trying to say. I didn't tell you how to get there yet. But maybe, let me show 2 more slides and then you can ask me again. I want to say that when I'm saying explicit representation of shape, we talked, I think you asked about identity and category. We assume that, again, category and identity are just different grains of shape discrimination. So you can do identity, this is actually subordinate level, which is what we were talking about earlier, right? But you couldn't imagine this is a boat, this is a car. It's basic level discrimination. But the concepts are still the same. You think you need a shape representation that can support these kind of things. Okay, so I want to point out that if you simulate this, just simulate this on what's going on in the retina, a million-dimensional space. This isn't quite a million-dimensional space. It's only like a 20-some thousand-dimensional space. With these 2 3-dimensional objects, and then you project it into a 3-dimensional space, it looks really awful, which is what means this is why it becomes hard. So these are objects undergoing view changes in this case here, and it looks like a red and blue thing like they're merged together, but they're actually just like crumpled up sheets of paper living in a high-dimensional space, and you can't easily see where the separating hyperplanes live. When we project it to 3 dimensions, it looks ugly, and we call that tangled. It's why you can't put simple linear classifiers on pixels to deal with recognition problems here with a limited number of training examples on pixel spaces. You need them to be in a much nicer looking representational space. Again, this shouldn't be news to anybody who thinks about feature spaces and machine learning, but I think for neuroscientists, you need to understand that mapping. I hope it's not news to you guys. I like to call this that the manifolds are tangled in this initial space. That's why it becomes hard and interesting. But so you have this tangled information, you think about state spaces and pixels, the retinal ganglion cells, and then you go through some sort of transformation, and we think you have some more untangled information. And I use that word to mean a couple of things that I've showed here. So first, that you have the separability, okay, that you can put a separating hyperplane between that, and then also, and you can do with lowish numbers of training samples. And then also, I think the word helps imply that it's not just building grandmother cells for Joe and Sam, right? Those would be like points in this space. Now, this is IT, right? So these are IT neurons here. This is how we think about IT. It's not just, there's a Joe cell and there's a Sam cell that live up in IT. Those would then those manifolds from the initial space would project a point in the space. So what are the other axes there? Does anybody know from what I showed so far? This is the shape axis I'm calling. If I put a classifier here, I can say that's Sam and not Joe. What are these? Can you see from what I went through quickly? What information is represented along here? Pose in this case, right? So we think there's other latent parameters that were there initially. They didn't throw away. We just recoded it. So it's here. And if you want to do other things like the person looking to the right, in principle, you can still have that data to some degree. And we've done some recent work on that if you want to talk about that. But I think this sort of gets people away from thinking about grandmother cells and just think about what are the latent parameters, the latent sources, and we think IT's done a reasonably good job. Let's say the ventral stream has done a reasonably good job, reflected in the responses of IT, of untangling those variables from each other.
Jim, can you go back for a second?
Yeah.
How did you get to the first, the crumpled one?
How did we generate it?
Yeah.
So this was generated so what you do is you take the 3D objects, you simulate them over, I'm trying to remember, this is pretty old now, it's at least 3 degrees of pose. I think that's what it was. Maybe actually in this case it was just 2 degrees of pose. And then you have all this points in a high-dimensional space, you simulate all that. Then you try to find the axes from, we were trying to find actually, even some training examples, what's the best way to separate. We had to come up with a way to give you the 3 projections that we're going to show you. And I'm trying to remember exactly how we did that. It was either a simple linear classifier or some sort of PCA. Which are close, not exactly the same.
Because, there are simple arguments that if, for instance, the transformation is a group, then you don't have crowd. Okay? You don't have intersections.
Well,
they are separate in the very high-dimensional space.
I'm sorry, thank you I didn't mean to imply these were intersecting. They look like they're intersecting.
Okay.
But that's why I went to tangle, because if I took 2 sheets of paper here and rolled them up, they never fuse. Except when these 2 people go to the back of their head and then I have ambiguous information, then they will contact. But we think that's the unusual case, I think, as you were saying too, that in the high-dimensional space, the stuff is still there.
Well, there is a lot of room in a high-dimensional.
There's a lot of room, but that's back to the point of you need to look. There's a lot of room, but finding that room with a limited number of training examples is still a hard problem.
That's it.
Yeah, so I I know there are 2 different ways to think about this. It's not, I think Tommy's point is not just about linear separability. In a high-dimensional space with enough training examples, I can find a hyperplane. Right? I think that's the fundamental point.
Yeah.
That it's there, but it's just very hard to find in that space. So another way to think of this is you rather think of it as not the linear separability is what's improving, but it's like the dimensionality of the space, meaning the number of training examples that you need to find a plane is being reduced, yet we're maintaining the information. So,
the IP is a recurrent network, or is it just there?
I haven't yet shown you evidence that IP does what I said here. I was trying to give you a sense of why the problem looks hard on the retina, and how you wanted to look at places like IP. There's some evidence to support that, of which I'll show you some in a moment. And some of that was previous that I briefly went through. But those, if you want, when I'm making this plot, when I plot this, all I need is, this is like the firing rate of one IP neuron over some time window. So, is that produced by a recurrent network? Possibly by this recurrent network, but we can measure the firing rate over some time window, and that's neuron 1, neuron 2, neuron 3, and then this is what the information looks like when measured that way.
Because then you need another dimension for time, to return time and some information in.
Right. Again, in my presentation, been ignoring predictions of estimates of time. We're just saying, here's an image, tell me what it was. And it's not about predicting what's going to happen next. It's just telling me to separate Joe from Sam. Core recognition is just a static image. And
static image couldn't have spatiotemporal.
It could do this, but I can show you the plots of how IP supports this, and actually the IP responses are pretty stable over the time window of 0 to 200 milliseconds. There's a little bit of movement there, and some information comes a little earlier than later. But I'll have to show you the data on that. This is just a fixed conceptual ideas about what the problem looks like. This is the problem we're thinking about. So again, you may not like it or not, but I think this is a discussion that's useful. And why is the problem hard? What would you want it to look like at the end? And now, what's the evidence that it looks like this?
Why do people in your lab, or why do some people in your lab not like the geometric stories? Can you make some offhand comment about that?
It's hard for me to say. I'm like this point of view. I don't know.
Sorry,
every time.
Story?
Alex was asking why some people don't like this point of view, and I actually can't. They just say, I don't like that. But the guy in my lab who's a mathy guy, he doesn't like it. Maybe because they're not real manifolds, there might be something who's there. But every time he describes what's going on, I think, well, what you really mean is that I can map what he's saying onto these words. So it's just a matter of how you like to think about the problem. I don't think he disagrees with it. It just makes it. I like to think about it geometrically, but maybe our intuitions for 3 dimensions to Tom's question are kind of not helping us to some degree. But I think, again, for the neuroscience audience, I think there's key things here that the problem is hard. This is why it's hard for the transformation variables, and that you don't throw everything away. And again, people have said that on the computational literature. It's just the field of neuroscience is sort of, they kind of don't know that or they lose track of that. So I think this kind of drawing helps bring a larger audience into what's going on and what would you want it to be. Yes, for this audience, maybe the details start to matter, but this is why we did this, was to explain to people what's interesting and what's hard and what the answer might look like. Just conceptual. Yeah.
Can we revisit this assumption that we need a linear separability?
Sure.
What evidence is there to support that?
Just the fact that you can do the times.
Why not anything more complicated?
Okay, so here's the way that I think about it. Because we're recording here, we want to ask about, we think these are powerful features, and then we do this. But of course, you need linear separability. But of course, if I was here, this is not linearly separable, which is exactly what's kind of being implied there. So I'm not saying a transformation is linear. I'm saying from some point in the brain, it's probably linear. And we think that point is here.
Well, why do you need linear separability?
Somebody, some downstream neuron has to eventually make a decision, whether it's a motor neuron or a basal ganglia, somebody has to make a decision. And that decision is modeled by a linear discriminant is one way to model a decision. Now, there are other ways, but they're all relatively simple. So at some point, you need to get simple. The motor neuron is not complicated, right? You have to get simple. So you have to make the information explicit with respect to the task somewhere in the brain at some point when you're doing the action. That's all I'm saying.
So the idea is that like the ventral stream is providing sort of like a modular interface to.
The ventral stream is providing a shape basis, which then could easily, let's say, downstream neurons. Like Monkey might not know who Joe and Sam is. I'm going to train you for a while on Joe and Sam. Oh, let's just learn up some linear weights, reading IC, problem solved on that linear performance. That's how I think about it. That's a hypothesis, right? I mean, you have, I'm going to say, you have a set of tasks, you have measurements, you try to ask what's the mechanism for A to B. So
I also had trouble with the linear thing, because it seems like, there are all sorts of classifiers. You can use whatever classifier you can.
Sure, and if we open a box of classifiers, then we can say, well, any classifier on the retina, throw all this brain stuff out, make this all a big black box, and say, I need a nonlinear classifier to the problem of separating Joe from Sam over a bunch of transformations, and it's a nonlinear classifier. Let's all get back to work. That's not helpful, right? Because if you're going to go in the system, you want to start to point and say, look, if it looks linear from here, now the problem's over here. If it's not linear from here, then the problem's over here.
So because you can use a linear classifier to decode at the final layer doesn't mean that the brain is or needs to do that.
Fair enough. But it's a when I remember at the beginning, it's like science is a predictive model. This is a hypothesis that's testable. If you have an alternative one, just put it up there and we'll test it. So that's kind of what we're doing. I'll show you the next part. Is like, if this is right, it makes predictions about what behavior should be like. So we go measure those predictions. And I'm not saying the brain should be linear, of course. It's like, I'm just saying from here to the kind of task that I showed you at the beginning seems to be well fit by a linear decoder. Now you can make, again, under the data, just like I say, you can make inferences from, well, I don't like the way you're thinking about the brain. I don't like that. That's not a real criticism to me. What I like is, here's an alternative way that is to also explain the data. So the data are the behavioral performances on the task in this case. And I'm going to show you that if you just do linear decodes on this, you can basically predict that. So does that mean the brain is linear decodes? I don't know. But it says it could be. It's a hypothesis. We're not going to prove anything. We're just going to test ideas. And so then we'll say, well, what does that predict that we haven't yet tested? What other idea would you like? So that's how we're proceeding. And I don't want you to think that's only linear, because this is not linear up here. This is linear, this is a rotation in space. I wouldn't have to do linear, just going to rotating that space around. It's got to be something more than that. That's the actually interesting part of the problem. All we're trying to do is say, look, this problem is already sort of solved up here, and then our work is mostly trying to understand what's going on in these nonlinear parts. That's how the lab's proceeding. We kind of started at the top to define something, and we want to work through the middle, understand how you produce that. Yeah.
So if the intermediate layers are doing some nonlinear transformations in order to pass the information along, why would you not have that same thing going on at the decoding?
Okay, this is a subtle point. We say this is linear, but it's actually linear nonlinear, right? Linear with a threshold. That nonlinearity, that's a huge nonlinearity, right? That's the models of this is also very much like that. Linear with a threshold. There's other types of nonlinearities, but linear with a threshold is nonlinear. So these are old ideas. We're not saying this, but when I say it's linear, I mean linear with a threshold. The decision is a threshold, not a nonlinearity. So I like the spirit of what you're saying. Like, this is tissue, whatever mechanism pops in down here. You'd like to think of it working here and here and here, whatever that class of mechanism is on. And linear with a threshold is a class of mechanisms. And I know it's ignoring the feedback connections. What are they for? We're just driven by the data to explain the data. And this is the fun discussion to have.
Yeah, just another comment on that. Even if you're using a nonlinear, kernel or something, eventually you're just going to use the linear separation. So if you're doing like a nonlinear SVM, it still boils down to SVM and just transform it into a space. So at some point, it has to be kind of linear. If you're...
Well, I think that's all I'm saying.
No, exactly. I'm agreeing. I'm just trying to explain.
No, I know that, but you can write any function as a combination of any other function, as two other functions. I mean, that's fine, but that's not really the point. Like,
yeah, I mean, of course, we all believe that global transformation is interesting and nonlinear. The question is, how do we get into the brain and make progress on what the brain is doing? And all I'm saying is go in and report IP. It looks like from that point, linear with the threshold explains the behavioral data in the task domain that I've defined for you. And your reaction to that might be, well, then your task domain is wrong, because I wish that it was nonlinear from IP or from some prior that motivated you to not like the work. But you can't deny the data that's
it's not that I wish it were nonlinear. I just don't see why the fact that, like, you could train other nonlinear models, classifiers on the IP data, and you would also be able to decode, right?
Yeah, but why, now we're going to think you like prior, we'll revert to parsimony and say linear is more parsimonious than those models. So I want the most parsimonious model from IP to explain the data. And that, so far, is...
I mean, I think it's sort of arguable whether it's more parsimonious to say, like, well, I'm going to go higher, so you use linear, than to go, like, why did they actually use a more complicated model?
Yeah, I thought you meant from IT. You were saying, given I report an IP,
I did mean that, yeah.
Given I report an IP, I can do linear decoders and nonlinear decoders. So I'm saying linear decoders work. And so what the inference we draw from that is IP kind of looks like this in its state space. That's what I'm saying.
You know, that is what I meant, but like, I mean, you could presumably go to a lower area and use a nonlinear decoder, and maybe that would also be parsimonious. I don't know.
Well, yeah, but you'd want to build that nonlinear decoder that's structured to look a little bit like this. But yes, you could do that. And if you want to go as far as you want to build nonlinear decoders to do recognition, you might, yes, you could model V1 right now. I mean, I can give you a model V1, and you can put, and that's in some sense what I'll show you at the end of the talk, what we've been doing on the encoding work. We're building models that go from here to try to solve the task, to try to find the nonlinear transformations, and then later ask how well do they look like the neurons. So that's just for us. We put the split here because we recorded here and call that MyLab bias. This is decoding and that's encoding. So we can use linear decoders from this point with a threshold, and we need nonlinear encoders to get to this point. And overall, the thing's nonlinear, but that's just where we key into the problem. And that's all. I mean, we could put this point here, we could talk about sort of linear here, and then build some nonlinear there, and just call that decoding, that encoding. But just relative to where you're standing, you know, it's going to be more or less nonlinear. That's all I'm saying.
I like the motivation of like not the like linear readout from like a simple model of a neuron with like a dot product and a threshold. Like that's a bit of a simple model of a neuron. Like we know that they're more complex in certain ways. So like what kind of classes of classifiers does that open up if you take into account like subcompartments? Yeah.
Right. I mean, even within the space of simple decoders, there's a large space of things. So. You know, at some point the data are not going to be constrained over all those possibilities. I mean, I'm not disagreeing with you any of this. But of course, the brain is probably not as simple as I'm describing downstream, but I'm saying data are sufficient to explain the behavioral performance, and that's a piece of evidence that we, as a field, have to reckon with and decide, well, what do we interpret that to mean? And all I'm saying is that this means to me that the layout state space looks something like this.
I think what Alex is asking, and what I've been sort of alluding to, is that like, as far as I know, there's other data that's well established that you're ignoring.
Like on IP?
No, like that point neurons aren't really neurons. And
neurons are not neurons?
Well, sorry, point neurons.
Point neurons are not.
But like linear nonlinear models for neurons aren't really like. They aren't perfect models of learning and the feedback stuff. And I mean, you of course acknowledge they exist, but you say like, well, I'm not going to. I don't want to deal with that.
I'm just saying I don't need to make a more complex model.
But it's not more complex. Like I mean, he's talking about data and like. That is the data. Like the data is.
No, this is a concept slide. There's no data on this slide.
That's what I was just
actually show you.
No, I was just pointing at the slide as if I thought that was actually the data. But the neurophysiological data, I think, are that there's like a lot of feedback, and it's become more complex to postulate that it's there because you know it's there.
Well, again, I'm not, this is, as I tried to point out, the fact that we can record up here does not deny the importance of feedback, if you want to use that word. So we have to define the word, but does not deny that. It just says, from here, you can get to the performance. If you like that, that's all it says.
I don't see how I mean, your model explicitly, or not explicitly, your model doesn't have that in it. Like,
Well, when you say my model, this is not, there's no model yet even on the page. This is just a conceptual idea, that here it looks pretty simple to do these tasks. And I'm going to show you the evidence that it works. So, that's all. I mean, why don't we just go through the evidence and then have this discussion again, okay? Because I think we should take a break soon, but I think I was near the end of this section anyway, so. I think, let me put it back up here. I just want to say for the kind of people that, many of you guys are computation oriented, I just think of this as a poor encoding basis for these kind of tasks, and this is a powerful encoding basis. It doesn't say how we got here, you can call it magic. The magic basis does well, not, that basis that we know how to build from the image does not do well. That doesn't tell you how to get to the magic basis, but it says magic basis is pretty powerful with respect to the task. And I don't mean powerful in a computer vision high-performing sense. I mean powerful in that it matches the kind of errors and difficulties that humans have. Which shouldn't be at all surprising, even unless you're a dualist, because the human has some neurons in its head that somewhere have to correlate with its performance. And that's, in some sense, what I'm saying from a neuroscience perspective. Those neurons, for these tasks, live here, and none of the quantitative evidence that I'm going to show you next. So let's take a break. Question. So I'm going to try to have a quick break, because this is actually where most of the data that we're discussing is here. And then if we have time, we'll talk about the encoding models, which is probably really interesting in this group. Yeah. Okay, one question.
I wanted to ask an historic question. Charles Gould wrote the history of grandmother cell, and it's like the concept was almost there already in Wiesel and Charles Gould, and the technique of single units too. So how come, it's a kind of a problem where both the techniques and the concept was almost there, and you could do all these experiments that has been done with single units in IT and showing images and show that. Why it took it so long to have these?
To do what? To do long? I showed you the history of the field. You record, you measure some images, and then you plot plots, and you try to make some inferences. You could view our work as part of that chain. But what step are you saying so long to do?
I don't know, because many work in neuroscience because of techniques evolve and MRI, and this seems like could have been done a lot earlier.
Well, yeah, really what this is just a blending, and I think Tommy and I did the initial work on this together. Tommy gives a lot of credit for it. This is just a blending of ideas from machine learning with population recording methods in high levels of vision. The ventral, a lot of people think vision again came out of a psychology background, and they were just like, I'm going to record a neuron, and all they knew how to do was to then try to show images and then guess what the features were. But if you start to take a different view of, let's think about the task and how you might solve it and what kind of decoders, the discussion we're having, then yes, these things kind of become obvious. I think that's your question, why weren't they done? I think it's just an issue of the people that were working these areas were pointing out this is a neglected part of the brain for the last 10 years. There's a Japanese group trying to largely neglected. So we're trying to bring ideas that a lot of you guys know, and some often better than I do, onto these kind of problems to map that on neuroscience. And I think that's the spirit of part of the CDMS effort, is like there's some insights here from both that inform in both directions. What are the neural data constrained on the modeling? What does the modeling tell us about what's maybe going on in the brain? And that's sort of, I think, the spirit of CDMS effort is about, at least, the vision part here as I see it.